Welcome everybody. So I hope you had a nice afternoon. As I've been doing all along, I'm not going to say very much about the speakers because I think their CVs and what they will talk to you about speaks for itself. But I do know of David's work for very many years, and I've always been very impressed by it. There are several of David's papers that I do point my students to regularly, and I just want to hand over to David Boas from Harvard, who's going to talk about near-infrared spectroscopy in the brain. So we're getting into functional imaging now. We started off with microscopy, ballistic imaging with OCT. You found out why it's not possible to do microscopy deeper inside the tissue from Igor Medlinsky with these tissue optics. And then we found that one solution is to throw away all those scattered and confusing photons with OCT and pick out just the ballistic ones. But now we're moving into, I think, diffuse optics. So thank you, David, for lighting up with that.
Thank you, Martin, for the introduction, and also bringing me to Ireland, my first time. I'm really happy to get to see the beautiful landscape. Actually, one of my dreams has just come true, where I just walk, on the burn. So really happy to, check off one of those bucket item lists. We have lots to go through on the diffuse optical imaging and near-infrared spectroscopy. Really focus on imaging brain function. That is what I'll focus on. The first half is really just going to be background and theory and the early applications from the 90s and early 2000s. And then the last half will essentially be the functional brain imaging from the last 10 years. And I think in between them, or whenever is appropriate, we will have dinner prepared by Igor, I guess. I know certainly with dinner comes drinks, and so I very much look forward to the drinks continuing during the second half. And if anyone is clever enough to figure out how to get drinks here earlier, that's completely fine with me, because you have to have your before dinner drinks. I don't know what the custom is in Ireland, but in Italy, that's what we always do. So if anyone is clever enough with that, please share. Okay, I have broken from the custom. I didn't prepare slides telling you about where I'm from. I feel really bad. So I'm from Boston. I'm at Mass General Hospital. And I wish I had, there's one perfect map of you can look at it, Google Maps. You see Boston, you see Charlestown, where I am, which is right on Boston Harbor, which is a beautiful place. Boston Harbor, nobody really knows about Boston Harbor, but there's dozens of islands out there that you can take a boat out to. There's even a national park out there. It's quite great. So if you're ever in Boston, make sure you do that. But I'm in Charlestown, just across the river from the north end. And then very close by is MIT, and a little further out in Cambridge is Harvard. And if you just cross the river back into Boston, then you got Harvard Medical School as well. So we have this just amazing density of great schools and great medical schools and great science. So I've been very fortunate actually to be living and working there for the last 20 years. So please come visit anytime. So let's start off with near-infrared spectroscopy, diffuse optical imaging of the human brain. Why would you do optical imaging of the human brain? Well, it complements the traditional methods, so you've probably, have you all heard of fMRI? Who's not heard of fMRI? Good, you've all heard of fMRI. So that, actually, one of the places it was really developed, invented, and pushed forward is where I work at Mass General Hospital at the NMR Center, formerly known as, and currently known as the Martino Center for Biomedical Imaging. They did the first fMRI experiments published in Science, but that's really kind of revolutionized this field of mapping brain activity. Before fMRI, there was actually PET measurements, positron emission tomography measurements of brain activity. It requires radioactive nuclides, so it's not as often done, its temporal resolution is not as good, so fMRI has really kind of blown open the door for brain mapping. EEG, of course, was done for a long time before PET and fMRI to measure the electrical activity of the brain. Has very good millisecond sort of temporal resolution, but its spatial resolution is really somewhat limited. And magnetoencephalography is the magnetic equivalent of EEG. It's measuring the magnetic fields and changes in magnetic fields induced by the electrical currents in the neurons. So near-infrared spectroscopy complements all of this on a spatial and temporal scale. Its spatial scale is not as good as fMRI because we're using light that is diffusing through the scalp and skull, so that diffusing photons lose their spatial information. But the temporal sensitivity really spans several orders of magnitude. You know, from sub-second signals to measuring signals over days. Systems are lightweight. You can wear it for days. But it also offers novel contrast mechanisms. So EEG and MEG are measuring really the electrical signals from the brain. fMRI is just measuring deoxyhemoglobin, which is paramagnetic. And so it measures the deoxyhemoglobin changes during brain activation. And PET, it can measure hemodynamics as well as glucose metabolism. With optical imaging, we have invasive methods, we have non-invasive methods. You wouldn't really do the invasive methods in humans. Those are primarily for animal studies, unless you want to volunteer having a window put into your skull. But you can measure neuronal signals, metabolic signals, and hemodynamic signals. Really, today we're just going to focus on the non-invasive measurements that you can do in humans. And so the most common, really what we're going to focus on today is just measuring the hemodynamic signals that are related to brain activity. And in the second half, I'll actually go into more detail about how the hemodynamic signals are related to neuronal activity. Okay. So this is the first half. We're really just going to spend a lot of time on the background of near-infrared spectroscopy and review some of the early applications. And if there's any questions, you know, feel free to ask questions and answer them if you want as we go along. Okay, so near-infrared spectroscopy works because the dominant chromophores in us tissue are hemoglobin, oxygenated hemoglobin, deoxygenated hemoglobin, and water. This is the absorption coefficient on a log scale. So you can see hemoglobin absorption drops dramatically as you move from blue light to green light to red light. And as you go into this near-infrared region, the absorption length of blood in us is on the order of 10 centimeters, right? So light can travel 10 centimeters through tissue, and only 63% of the light will be absorbed. Okay. Now, in laser pointers, you know, about 10 to the 18 photons per second. We can detect hundreds of photons per second with a good SNR. So light can travel meters through tissue and we get good, good photon candidates. If you go more into the infrared, water absorption goes up. Light doesn't propagate very far because of the increased absorption. Looking on a linear scale now, you can see this window, this optical window, is much more apparent between about 700 nanometers and 900 nanometers. This is where the absorption is very weak. Light can travel great distances through tissue. The problem is that the scattering length is much shorter. So the scattering length is actually much less than a millimeter, but the scattering is really strongly forward directed. The random walk length, so the distance that a photon travels before its direction is randomized within tissue, it's really on the order of a millimeter. It's kind of on that scale that photon transport through tissue actually follows a diffusional process because its probability of its direction being randomized is a much shorter length scale than the probability of a photon being absorbed. So because that absorption is so much weaker than the scattering, light is diffusing through the tissue and can actually diffuse through the scalp and skull. So early on, when I first started presenting these ideas to audiences 20 years ago, people always said, well, light can't penetrate the scalp and skull of a human. So we had to get a three-dimensional model of the human head and run a Monte Carlo simulation of photon transport through the human head. So we just assigned typical optical properties to each tissue type and did the simulation, and you could see, sure enough, the light is going everywhere through the head. I should point out, I'll stop here, this color map is spanning about 10 orders of magnitude, right? So the light is propagating through the tissue, but it's attenuating quite strongly. So if there is no scattering in the tissue, in 0.7 nanoseconds, that pulse of light would have traveled in a straight line, to about here, about to the neck. But because the scattering is so strong, the fluence of photons, not uniformly, but disperses throughout the head. Okay. So looking at this in a little bit more detail, we can calculate just the distribution of light. If we have a source here, the light will be distributed throughout the head. You can see most of it is near the scalp and skull. Some of it reaches down into the brain. If we have a detector displaced by a few centimeters, this is the probability of light reaching from some point in the tissue to the detector. And if we take the product of these two, this is what a sensitivity profile is going to look like. If we have a source and detector on the head, this is the sensitivity we would have to absorption changes in different parts of the head. So we're, as you can see here, and we'll come back to this more frequently, we're most sensitive really to the absorption changes in the scalp and skull. But we do have the sensitivity to the cortex underneath the scalp and skull. So these measurements, these near-infrared spectroscopy measurements of brain activity really have their origin back in oximetry. And people first started to use light to measure hemoglobin oxygenation in the 30s. And it was several decades of efforts that were not very reliable. It wasn't really until the 70s when there was a clinical breakthrough with pulse oximetry. So prior to pulse oximetry, they were shining in many colors of light into the earlobe, or the fingertip, and they were trying to measure the characteristic absorption spectrum of hemoglobin to differentiate oxygenated and deoxygenated hemoglobin. The problem was they were always confounded by scattering, and they needed some way to calibrate scattering, because the scattering would change the path length of light through the tissue, and the scattering was wavelength dependent, and they didn't really understand what that wavelength dependence was and the impact it would have on the path length, and so they couldn't ever really accurately calculate the oxygenation. But with pulse oximetry, it actually introduces this pulsatility onto the absorption coefficient. It's pulsating the amount of blood in the tissue, which pulsates the amount of hemoglobin, which then pulsates the amount of light we detect. And they, just through some clever algebra we could do tonight after some beers, you can actually calibrate out that path length effect and retain the hemoglobin oxygenation. So this is the basis, really, of everything I do, is this hemoglobin spectroscopy. But what NIRS does is really solves that problem of what is that wavelength-dependent path length of light through tissue. Okay. So near-infrared spectroscopy, wanted to extend beyond pulse oximetry, solve that problem I was just telling you about. Jobs published the first NIRS paper in 1977, showing that you can actually measure hemoglobin changes in the human brain using near-infrared light. It was very quickly put into clinical studies in the 80s, and it was in the late 80s, 1990, that Mark Cope and David Delpy really kind of spelled out the quantitative math behind near-infrared spectroscopy, which is incredibly simple. And if you remember nothing else tonight, this is the one thing you need to remember, is the modified Beer-Lambert law and how that describes the NIRS signal. So the Beer-Lambert law is just describing the optical attenuation of light that you get if you shine light through a solution, some absorbing solution, no scattering, just some absorbing solution. Very standard chemistry, spectrometer type of measurement, where you have a test tube filled with some solution, you want to know what's there, and you just measure a spectrum of it, and you measure the optical density of the light, the wavelength dependence of the optical density of the light, how much it's attenuated, and that's then related to the concentration of the chromophores in your test tube, times the extinction coefficient, the intrinsic absorption and extinction of the chromophores, times the path length of light through the test tube. Okay, so all you need to know is that path length and the extinction coefficients, which are intrinsic to the chromophores. Given these measurements, you can then solve for the concentrations of the chromophores. The challenge with near-infrared spectroscopy, though, is that tissue scatters light, right? So if we have a source, here and a detector there, while the net distance may be 1 centimeter, the total distance that light actually travels is dispersed, and on average is going to be 10 centimeters. Right? There's some photons that will be only just a centimeter or two, but many more that travel several centimeters because the scattering is happening on such a short length scale, and the photons are undergoing a random walk. So you have to put in these fudge factors. So you need a fudge factor, this differential path length factor, which multiplies the physical distance between the source and detector. And then you need a geometry term, because now that light is scattering, not all the light travels straight through the test tube. Some of it's going to scatter and exit out the sides, or exit out where it came from. So this geometry factor accounts for that. So we have two problems to solve. We have to know what this geometry factor is, and we need to know what this differential path length factor is. So Mark Cope and David Delpy said, well, you know what? If we just want to measure changes in the absorption coefficient, the geometry factor doesn't really change if I have some local change in the hemoglobin concentrations. So if I measure a change in the optical density, G cancels out, right? So now all I need to know is what this differential path length factor is. And so then, it's actually, today we kind of know what those differential path length factors are. And so we can just take very simple measurements of the change in optical density due to hemoglobin changes in tissue, and then relate that to changes in oxygenated and deoxygenated hemoglobin. Very simple. So this is Jobs' first experiments. This was in a cat, and making measurements at two wavelengths, 760 nanometers and 815 nanometers. And a cat was anesthetized and on a respirator. And so they stopped the inhalation of oxygen. And you can see basically the light signal is going up, indicating that the hemoglobin is becoming more deoxygenated. At 815 nanometers, this is really a measurement of total hemoglobin concentration. At this wavelength, the extinction coefficient of oxy and deoxyhemoglobin is pretty much the same. So you're only sensitive to the total hemoglobin concentration, which goes up only slightly, right, to compensate for the fact that there's actually less oxygen in the blood. So then finally in that paper he went on and showed you could actually make a measurement on a human. This was a human that was hyperventilating. When you hyperventilate, you actually expel CO2 from your blood, and when the partial pressure of CO2 drops in your blood, your cerebral blood vessels actually constrict. So the blood volume reduces, and so because the total hemoglobin concentration was reducing, the light propagating through the head actually increased. And so that's what we saw here, was this increase in light propagating through the head. So those were the first measurements on humans. Right. So this modified Beer-Lambert law, let me now just delve into a little bit more into theory, right? So this was just kind of written down ad hoc. It made perfect sense, but there is fundamental rigor behind this. We could go back to Maxwell's equations to drive the modified Beer-Lambert law, but we're not going to do that. I'm going to ignore all of the coherence properties of light because tissue is a very random medium. We don't have to worry really about the spatial modes of light and the interference patterns. We can just really treat the electromagnetic field as photons that are bouncing around like billiard balls or drunken sailors through some highly scattering medium. And so we can just use the radiative transport equation to describe that. And I still love this equation. I think I was quite intimate with it when I was a graduate student, although I'm not so intimate with it anymore. But it's just a mass balance equation describing the irradiance of light propagating through the tissue at a particular position, in a particular direction, at a particular time. And the key component is this term here, which accounts for light that scatters from one direction into another direction. Now, we're very fortunate that the probability of scattering is much greater than the probability of absorption. And because of that, there's so much scattering that this irradiance of light, the angular distribution of that irradiance is nearly uniform. And we can describe it really with just two spherical components. The isotropic component is called the fluence, and the first moment, angular moment, is just called the flux. Right? So that's all we need to describe the irradiance of light in tissue. And so when we actually do this approximation and reduce the angular moments just down to these, the 0th moment and the first moment, we arrive at the diffusion approximation. And so that's another thing which maybe I could still do, is actually go through that derivation to go through the radiative transport equation, and you arrive at the diffusion equation. And you can write down the diffusion equation, either the time-dependent diffusion equation, or just say my fluence is going to be oscillatory in behavior, so my time derivative is just going to have a frequency, so I can just write down the frequency domain diffusion equation. And so we use both of these actually to describe the propagation of light through tissue. So the solution of the diffusion equation, for the time domain, we could put a pulse of light into tissue. Some photons will travel relatively straight through. Other photons are going to take a long, tortuous path through the tissue. The solution of the time domain diffusion equation, given by this, describes that temporal point spread function quite well for a highly scattering media. We have the speed of light, the absorption coefficient, and D here embodies the reciprocal of the scattering coefficient. In CW, or the frequency domain, the solution is even simpler. It actually kind of has the form of a spherical wave that's propagating outwards from your source that is exponentially damped, actually, because this K here is complex. And I'll come back to that in a minute. So, so that's all nice theoretically, but experimentally you have to make measurements of these signals. And the vast majority of people doing near-infrared spectroscopy are making measurements with continuous wave systems. So they just have a a laser that's on constantly. These are very inexpensive systems, relatively speaking, and very fast because you've got lots of photons. Detectors are sensitive and fast, and you're just measuring amplitude. At the opposite extreme are the time domain measurements. These are very expensive because you need a pulsed laser that needs to be picosecond pulse of light, and then you need the even more expensive detectors that can resolve this temporal point spread function that is typically broadened out to 1 or 2 or 3 nanoseconds at most. So, and you really want to have better than 300 picosecond temporal resolution of that temporal point spread function, if not tens of picoseconds. In between, you have this nice happy compromise, frequency domain. So you can actually just modulate your laser at 100 megahertz, right? And that allows you don't get the full temporal point spread function, but you get one frequency component. It allows you to measure not just the amplitude of the light propagating through the tissue, but you get a phase delay. So that phase delay is really important because it tells you something about the scattering and how it delays the propagation of light through tissue. And that's really important for helping us disambiguate the effects of scattering and absorption on the amplitude. If you just measure amplitude, you can't really distinguish scattering and absorption, right? Because if either one goes up, your amplitude of light is going to go down, right? But if scattering goes up, it's actually going to cause a greater delay in the propagation of light through tissue. Whereas if absorption goes up, it kills those longer path length photons, so you actually get a decrease in the phase delay because you're biasing the measurement towards the shorter path length photons. So that phase is really very useful, and making a frequency domain instrument is much easier than making a time domain instrument. Okay, but the first instruments made were CW. The second instrument that was utilized was a time domain system. David Delpy had a nice relationship with Hamamatsu and lent him a very expensive, particularly at the time, streak camera. And he had a pulse laser, and so he was able to use this very cool streak camera to measure this temporal point spread function. And basically what he did with that temporal point spread function was to measure the average path length of light through tissue. So he made measurements with different source detector separations. He got what the average path length of light through the tissue was, and he was able to use those measurements to figure out what this differential path length factor should be so that they could use their simpler CW measurement and analyze changes in oxygenated and deoxyhemoglobin, right? So they weren't going to use this system in clinical measurements because you needed 3 physicists to operate the instrument. So they used it to really calibrate the simpler CW system. Okay, so, and then at the same time, actually, Michael Patterson, he wrote down this time domain solution of the diffusion equation and showed that he could describe very well the experimentally measured point spread functions, and he could accurately estimate the absorption and scattering coefficients. It was funny, forever I knew this as the Patterson equation. It took me many years for me to realize, oh, that's just the time domain solution of the diffusion equation. Another interesting little side note, this paper is cited 1,500 or 2,000 times. So it's a real landmark paper. So I started in grad school shortly after that, and with Peyton Chance, and he was very interested in these frequency domain measurements. So Maureen O'Leary and I were new graduate students, and our first task in the lab that summer of 1992 was to measure the theory showed that there should be this exponentially damped wave propagating out from the source. So that was our first job, was to measure that. It was kind of cool to us. How could you have this random ensemble of photons diffusing through a scattering medium, producing, in effect, a coherent wave? It didn't make much sense to us at the time, but that's what the theory said. So we went out to measure it, and so we built this fish tank, filled it up with intralipid, which looks like milk, doesn't stink as bad. and we put a fiber optic in the fish tank, which was delivering light that was modulated at 200 megahertz. The intensity was modulated at 200 megahertz. And we had another fiber optic that received the light, took it to a photomultiplier tube, and we could measure the amplitude and phase of that modulation, the 100 megahertz modulation. Just move that detector to many different locations, and we were able to map out these contours of constant phase that were emanating outwards from the source. We thought that was incredibly cool. That, you know, it basically, theory predicted phase should increase linearly with distance, and that's what we saw. Theory predicted that the amplitude should decay logarithmically, and sure enough, that's what we saw. And you know, from the theory, the slope is related to the absorption and scattering coefficient for both the phase and amplitude. And Enrico Bertone at the same time with Sergio Fantini and Mariangela Franceschini, they had also built a frequency domain system. They were doing that multi-distance measurements and showing that using those slope information for amplitude and phase, you could quantify absorption coefficients and scattering coefficients. So now this is important because this is different. With a CW measurement, you measure changes in the absorption coefficient, changes in the hemoglobin concentration. With a frequency domain instrument, because now we have that phase information, it's giving us information about how long it takes light to propagate through the tissue. It now allows us to distinguish scattering from absorption and actually quantify the baseline properties, not just the changes in the absorption coefficient. And so that opens up lots of opportunities for additional applications to measure the baseline physiology of the tissue, not just changes in the physiology of the tissue. So how about the instrumentation? Right, so I've spent a lot of time building continuous wave instruments, working with one of my first students, Andy Siegel, who is an outstanding analog electrical engineer. We built our first breadboard system, and then I was introduced to a contract engineering company by Hill, from Horowitz and Hill, The Art of Electronics. I don't know if you know that Bible, but it was very nice. He came and visited our lab, and he looked at everything we were doing, and he said, "Oh, great electronics, so you want to make this bigger and better. You know, I suggest you don't hire an engineer, you contract 10 engineers," which was a brilliant suggestion. And he introduced me to this company, Tekken, where I was able to contract 10 engineers, and they took our breadboard, put it into a system, and then they made it bigger and bigger, so that now we have 32 lasers and 32 detectors all in this nice box. And better yet, because they can build it into a very nice box, it was very easy to then distribute it to other people around the world. And I think now almost 100 systems have been distributed around the world. So these
you say that with the frequency domain system you can distinguish between the scattering and the absorption, which you denoted mu-S and mu-A. Yeah. But what about the, what's it called, anisotropy of the scattering?
Yeah, good. Oh, excellent question. So the anisotropy of the scattering is not relevant because the irradiance is nearly isotropic, and the photon directions are randomized on the length scales of interest. We are essentially not sensitive to the scattering anisotropy. And that actually, if you go back to the radiative transport equation and make that diffusion approximation, you actually come up with a similarity relation. What you find is that the diffusion equation depends on mu-S prime, where mu-S prime equals mu-S times 1 minus g, where g is the average cosine of the scattering angle, right? So basically what that says and shows is in a diffusion, in a measurement which satisfies the diffusion equation, you cannot actually distinguish g from mu-S, right? So what we're sensitive to is the product, right? Which is why I was mentioning the random walk length at the very beginning. With these NIRS measurements, we're only sensitive to that random walk length, right? That's the distance a photon typically travels before its direction is randomized. And that's exactly what that similarity relation shows, is basically, as I make the G higher, so scattering is more anisotropic, more forward directed typically. And I make the scattering length, so basically when it's more forward directed, I need many more scattering events for the direction to be randomized. But if I make the scattering length much shorter, then basically I end up with the same random walk length. Okay. So we have this frequency domain system. One point to make here is, and I'll emphasize this a little more, we're actually modulating the lasers at audio frequencies, and we do that so that we can essentially frequency encode the lasers. So all the lasers can be on at the same time, and at the detector, we just have to do a Fourier transform and we can get signals from all of the lasers at the same time. So it's just a multiplexing strategy that allows us to get a lot more channels of data in parallel. It raises the shot noise floor, but we'd be so lucky if our measurements were limited by the photon shot noise. We're actually more limited by physiology in the tissue. Frequency domain systems, actually Enrico Berton with ISS and Mariangela Franceschini built the first commercial frequency domain system. So that's what the first prototype looked like, and there's the first commercial systems. So this system now also has 32 lasers. Typically, 16 at each of 2 wavelengths, and then it uses photomultiplier tubes for the detection and modulates at 110 megahertz. You can buy a commercial time domain system now, finally, after 25 years. Hamamatsu is now selling this time domain system, where they have pulse laser diodes, picosecond pulse laser diodes, and they're using photon counting photomultiplier tubes in order to measure the arriving photons and the time delay of the arriving photons. I don't think we need to go into the details of how that works. Basically, you've got this commercial system has 3 different wavelengths that the lasers are pulsed one at a time. It's time-encoded. They're fiber optically all put into the same fiber, which is delivered then to the subject. And then you have a single detector receiving the light, photon counting photomultiplier tube, and then it goes into these channels for converting a weak electrical pulse from the anode of the photomultiplier tube into a digital signal indicating the arrival of a photon. So it has to go through a constant fraction discriminator to clean up that weak pulse, and then a time to amplitude converter to then give you a digital pulse that you can then figure out the time delay of and histogram it to produce this temporal point spread function. So that time domain system, they call it time resolved spectroscopy system. So it's typically one source, one detector. Every detector, needs this circuitry behind it. So if you wanted to have dozens of the detectors, it will get expensive pretty quickly. We built in my lab a system. It was a time domain imaging system. So instead of using photon counting photomultiplier tubes, we actually used an image intensified CCD camera, and we imaged like 150 different fibers onto the image intensifier. So in that way, and the image intensifier basically acts as a gate. It could be a 300 picosecond temporal gate. So it only measures photons within a 300 picosecond gate. But I can do it with over 100 fibers all in parallel. Right? And for me to measure the whole temporal point spread function, what I have to do then is just change the delay of that gate on the image intensifier. And then I can build up a measurement of my temporal point spread function for more than 100 different fibers. So there are many practical measurement issues to consider with hardware. You always want the best signal to noise ratio. So we spend more money on more expensive detectors that are more sensitive. We want a high dynamic range, so we always have to be worried about that because the signal from a source is decaying exponentially. Right? So my signal 1 centimeter from a source is going to be about 10,000 times higher than the signal that's 3 centimeters from the source. And if you want to measure both, your detector better be up for that challenge of having that dramatic dynamic range. And of course, We are limited on source power. We can't put watts of tissue onto tissue because the tissue would burn. So we're limited to tens of milliwatts so that the tissue doesn't heat up in the near infrared. Of course, if you go to green light and blue light, you have to use even lower powers. But we would never do that for these measurements because, remember, the absorption is so high the light doesn't propagate very far through the tissue. Okay. Source encoding, I was alluding to this a little bit. We have to multiplex our lasers. We could temporarily multiplex them, which has the advantage that one laser is on at a time, and so the shot noise you get is only from each laser that is on. But I don't like that because of the reduced duty cycle. I, for these measurements, really prefer frequency encoding because I'm able to actually measure the signal from all of the lasers at the same time. Each laser is modulated at a slightly different audio frequency. And sure, my shot noise is going up because I have light from more lasers, but in our measurements, we've never found that to be an issue. Nonetheless, at the early days when we were first building prototypes, we really didn't know which would be best, so we built a prototype time-encoding system, so it looks something like this. That's a nice breadboard system, showing you how simple it can really be, and all it's doing is pulsing different lasers in sequence, different colors of light actually, and this is the signal on the detector, and then you can just lock into each one of those to get the signal for each wavelength. Schematically, frequency encoding would look like this, where you take your 4 lasers, modulate them at different frequencies, and then you digitize the signal from the detector, do a Fourier transform, and you can pick off the amplitude of each frequency component. So this is really the approach I've been using in our systems over the years. For the optical sources that you'd use, you can use laser diodes and light emitting diodes. I've always used laser diodes myself because I like that it has a higher power density. It's easier for me to focus into a fiber. It's also more narrow band than an LED. More and more people are using LEDs these days. Power density is going up. It still is hard to focus them into a fiber, but people are more and more these days thinking about making portable NIRS systems, and so they actually just put the LEDs right onto the head, right? And so that actually works quite well. So I think over the coming years there's going to be more and more NIRS systems that actually use LEDs. There's a curious advantage of LEDs in that you don't have to worry about laser safety. So I think many places they don't want to discriminate when you're doing human subject research. They don't discriminate between a laser pointer like this and a laser used for cutting tissue. They just won't make that distinction, and so you have to build your case that no, this laser pointer is not going to burn the tissue, it's not going to hurt. So you just have to build that case. With an LED, you don't have to do that, which strikes me as really bizarre because these LEDs today can have huge amounts of power and can burn the tissue. So they're not regulated as far as I know. So be careful about that. So optical detectors, there's basically 3 common detectors that we use: silicon photodiodes, avalanche photodiodes, and photomultiplier tubes. Pin photodiodes are really nice because they're very cheap. They're very linear and they have a very large dynamic range. Unfortunately, their sensitivity is not as great as a photomultiplier tube. But for more and more portable systems, people are using them because you can get the detector right up on the scalp, and they kind of work, particularly on the forehead where you don't have hair interfering with the signal. People are making inexpensive NIRS systems that use photodiodes on the forehead. I really like using avalanche photodiodes myself. They're very robust, they're more sensitive. The dynamic range is limited, but more limited than a pin photodiode, but it's still over 3 orders of magnitude and works pretty well for us. I, for CW systems, have not used photomultiplier tubes because basically they're a little bit more sensitive to room lights, and I wanted to put this in the hands of psychologists and such, and I didn't want them blowing up my detectors. So avalanche photodiodes were more robust for that purpose. I think I just said all of that actually. So, all right, let's go on to some applications. When do you want to do dinner? I'm only.
Good question. So,
I have 2 parts, so I'm still in my first part. A natural breaking point for me would be in like 40 minutes. So you're playing 7 for dinner. Okay, all right, so I think that will work out very well.
Okay, all right. So maybe since we've stopped, we could provide a few questions.
Yeah.
Yeah. The Biophotonics has come up with this concept of snake photons.
Yeah.
And I was wondering, how is that related to, of course, it's scattering and directional. So how is that related, or how can it help with this kind of imaging? This is my number 1. And my number 2 is, you mentioned that Hamamatsu actually makes those machines. Is it lab NIRS actually, is it related to that? Hamamatsu has a lab NIRS instrument, which is, you put the casket on your head, and then you have all those LEDs, or maybe fiber optics.
Okay, I understand. So snakelight, I remember in the 90s all this fantastic discussion about snakelight, which I haven't heard since 1999. So I think the reason I really haven't heard much of it since then is that there's just so few photons that are so weakly scattered that you can't make much use of it.
But still,
I think in a mesoscopic sort of realm, where maybe you're measuring no more than a centimeter of tissue or less, I think if you have applications for that, I think we should revisit, snake photons. But it will be driven by the application, right? Where you're imaging in this, millimeter to several millimeter length scale range, where you would have lots of snake photons. Right? But I haven't seen anyone present such a application that would use that yet.
You're still interested in that? You're still using some of these concepts in bone imaging, because again, you have very highly scattering areas.
I see.
Very
and then the other question you're asking about the Hamamatsu system. So, there are many commercial systems. So I think what you're thinking about is probably actually from Hitachi, not Hamamatsu,
maybe,
which is a new portable system they have, so that you can measure brain activity as they're walking around.
So how does that compete with this? Is it a competitive?
Well, so that's a continuous wave NIRS system, right? So it falls squarely within everything I was just describing as a continuous wave system, right?
Yeah. So
about halfway through, you showed a picture of how time domain light pulse then evolutes when the temperature of the medium becomes wider and lower. Can you just give an idea of the numbers, like give a few centimeters of tissue? How much does the pulse widen?
So he was pointing out, and this was, I wish I had a better figure illustrating this, that temporal point spread function, how is it modulated by absorption, how is it modulated by scattering, and with source detector separation, right? So, and what is the typical broadening? Actually, if you only go down 50% from the peak, the broadening is less than a nanosecond. Even for measurements out at 3 centimeters, the broadening is not much. But a good time domain measurement of the temporal point spread function, you're going to have good SNR down 3 orders of magnitude, right? So when we think of that broadening, you might go down to the 10% level of the peak, right? And you can see that tail going out. That can easily go out 3 nanoseconds. If I make absorption larger, the tail actually drops more sharply. And actually that tail, if you plot the temporal point spread function on a log scale, that tail is linear, right? And the slope of that tail is the absorption coefficient. Right? It's a very simple measurement of the absorption coefficient. And so as you increase the absorption coefficient, the slope just increases, and it decays more rapidly, right? If you increase scattering, that tail, the slope, doesn't change at all. What happens as you increase scattering, the rising edge of the temporal point spread function smooths out and takes longer to reach the peak, right? So that's what increasing scattering does, is it affects the rising edge of the temporal point spread function. As does increasing the source detector separation. So going to larger separations just smooths out that rising edge of the temporal point spread function.
Okay.
Yeah.
You said that you want to be interested in different sensitivities, like when there's a sensitivity difference in 3 orders of magnitude. And you also said that in a time domain system, you can only capture 1 photon out of 100 pulses.
I didn't say that. I thought it. Or this. Okay, the screen said it. Right.
I was just thinking,
yeah.
How does that relate to your acquisition time if you want to measure really?
Yeah. So that's a really important limitation with the photon counting time domain systems, is that you don't want pulse pileup.
Yeah.
And so what happens with pulse? So for every laser pulse, the detection system can only detect 1 photon, right? So if actually more than 1 photon arrives, you're going to miss the later ones, and as a result, you bias your temporal point spread function. You'll actually compress, right? Because you're missing the later photons. So the only way you can make sure you don't have that bias is to make sure the photon count rate is low enough that essentially you're detecting no more than 1 photon for every 100 laser pulses, right? So that just statistically makes it highly improbable that you'll have a pulse pileup problem.
Sure, but does it still resulting in a reasonable acquisition time?
So then to get a reasonable temporal point spread function that you can get a reasonable estimate of absorption and scattering, 100,000 detected photons is a pretty good target. So, a second is still quite reasonable.
Okay.
Right? Now, if you need to start multiplexing, then you multiply it by the number of sources you're multiplexing, right? So that just increases the acquisition time. Okay. Any more questions?
I was just going to draw a comparison there. You said that in going through the adult or A-brain, A-head, that you get down about 10 orders of magnitude in your photons, so 10 to the minus 10 or 10 to the minus 9. And that seems to me similar to, with OCT, we only see about 1 millimeter, because after 1 millimeter, we're only getting about 1 in a billion photons. Right. They're ballistic coming back. Those are ballistic photons. You get farther because you can use all the diffuse photons. But with both of those systems now, we're talking 10 to the minus 9. You need really 120 dB signal to noise ratio to make any sense of that, don't you? So you need
but we're not measuring the source power, right? So our systems, great questions. Our systems are always, our noise floor is around a picowatt, and our maximum signals will be 100 picowatts. Hundreds of picowatts will be our max. So our dynamic range is really kind of bracketed around that. So if it hits 10 nanowatts, we're already saturated with the detector.
What's the incident
for
those numbers? I'm sorry. What's the incident for those numbers? The incident laser.
So we're using, our human measurements, 10 milliwatts. Okay. 10 milliwatts, right? So great point. So 7 orders of magnitude. And I should have pointed out, right? So when it's 10 orders of magnitude attenuation in an adult head to go from the surface to the center. Be 10 orders of magnitude attenuation to come back out. So if what you wanted to do was measure the center of the head, you'd get 1 photon every 100 seconds, right? Fortunately, we don't want to measure the center of the head mostly. We're measuring the scalp, right, which is much closer to the surface of the head, and we get plenty of photons.
Keisha, another question.
David, how do you deal with artifacts due to fiber, scalp, or head variability to such a system? Because that must create some...
Well, so, the coupling of the fiber to the head will introduce artifacts, absolutely. But with brain activation measurements, we're looking at changes. So we're looking at the fractional changes in the signal. It calibrates out all of the coupling issues for those measurements. Now, if you want to do other measurements where you need the absolute intensity, then you have to worry about that, right? Absolutely, you have to worry about that. And so then you take great pains in designing a probe, that allows you to kind of get through the hair to minimize the coupling issues. You need to have redundancy in the measurements so that you can compensate for those artifacts, so that you know when those artifacts are happening, right? And so you can either repeat the measurement or throw out a subset of the measurements. So very important problem you have to worry about, except for brain activity measurements, we don't have to worry about it. Yeah.
And just one question, you mentioned that when you compare the time domain to the frequency domain setup, that the frequency domain setup actually allows you to do quantified tissue property measurements as you have amplitude and phase available.
Yeah.
But on the other side, even in time domain, you have the amplitude and the timing, and then over the Fourier transform is more or less equal information. So how does frequency domain help you give information which you can't have in time domain?
Well, actually, time domain has a lot more information because it has all the frequencies, right? No, it's just that, right, absolutely. If you had to choose one, you choose time domain. But if you worry about the expense of the system and how easy it is to use it, you choose frequency domain, right? It's just that frequency domain is a simpler system to build.
But you said there's quantitative information accessible in frequency domain.
Yeah, because it has that phase delay, right?
And you have the time delay, yes, phase delay in time domain too, or the timer.
No, absolutely. So the time domain gives you better information, but the frequency domain also has the time delay information. So the frequency domain has sufficient information to distinguish scattering and absorption. You don't need all the frequencies, right? You just need some information about the phase delay of light through the tissue.
Okay, this compared to the CD audio mode, we do not have the phase delay.
I'm not understanding.
Yes, was just right. So in the CV mode, you don't have any information to distinguish absorption and scattering.
Sure.
But the time domain instruments are quite complicated. So you usually won't get this to the clinic. You will. So it is possible, but quite hard. I have a system that does it. I won't put it to the clinic. It's for
the Hamamatsu's commercial system now is one that you could take, but that was 20 years in the making, right?
Yeah, and the frequency domain they're simple. They're as simple as the CD, just that you can differentiate scattering and absorption.
Okay, is that good?
Yes.
All right. I thought you were saying the frequency domain can't measure.
No, I thought that the slide where you said that unlike in time domain, the frequency domain allows you to measure quantitative.
It does.
Yeah, exactly. That makes sense. That makes sense.
Oh,
I'm happy.
All right. Okay, sure. Yeah, fine. Okay, where are we? Okay, so let's talk about frequency domain measurements some more. These are experiments that Mariangela Franceschini did with Dennis Huber and Enrico Grattan and the people at ISS as well. After they built the first frequency domain system, the grant reviewers kept telling them they needed to validate it against gold standards to show that they were really accurately measuring hemoglobin concentrations and oxygenation. So they did some experiments on piglets. Piglets are a good model because their heads are a lot like human heads in terms of the thickness of the scalp and skull. Not pigs, piglets. Pigs have really thick skulls. So if you're ever doing an experiment, don't do it on pigs. You'll get no signal. It happened to me once in China. I took a NIRS system to China, and piglet didn't translate. They prepped a pig, and we went in to make a measurement, and there was no signal. It was very sad.
Did you get a piglet eventually?
Yeah. Not that night, though.
After about 3 months.
Right. They put a lot of instruments on this piglet, so that they could get from the femoral artery measurements, blood gas measurements of arterial oxygenation. They also had, yes, a femoral vein so they could get venous oxygenation. They had a pulse oximeter to give them an optical measurement of the arterial oxygenation and heart rate. And they even had a strain gauge to measure the breathing of the animal. So you'll see why all of that's relevant. So the first experiment was just changing the inspired oxygen. And that, as you decrease the inspired oxygen from 15% to 10%, what they saw in the multi-distance frequency domain measurement, they were able to quantify absolute absorption coefficients and from that get absolute hemoglobin concentrations. They saw that deoxyhemoglobin went up, oxygenated hemoglobin went down, because basically the amount of oxygen in the arterioles has dropped. And since the tissue is using the same amount of oxygen, we're just going to extract more of that oxygen from the hemoglobin. And so that's why we see deoxyhemoglobin go up. Physiologically, because there's less oxygen in the blood, the arterioles actually dilate in order to deliver more blood to the tissue to compensate for the fact that it has less oxygen. And this is why we actually see that the total hemoglobin concentration goes up. So as for the gold standard measurements, so they measured, if you take oxyhemoglobin divided by total hemoglobin, it gives you the hemoglobin oxygen saturation, which is the magenta line here. And that could be compared against arterial oxygenation measured by blood gases in the femoral artery, venous oxygenation measured by blood gases from the femoral vein, and against, well, the blue one I won't talk about. So you can see that the NIRS measurement of the saturation, was bracketed quite nicely between the femoral artery and the femoral vein, right? And that makes perfect sense because the NIRS measurement is not measuring just arteries, it's not measuring just veins, it's measuring the arteries, capillaries, and veins. And so it should be a measurement that's really just a weighted average of the oxygenation of the arteries, capillaries, and veins, right? Now, if you were to cut some tissue and count all the arteries, capillaries, and veins, you'll see that it's a roughly less than 20% of the volume is arterioles, 40% is capillaries, or maybe 50, 60%, and the remaining is veins. So really, from a blood volume perspective, we're much more weighted towards the capillary venous oxygenation, which is why this tissue measurement is much closer to the femoral vein, right? Blood gas gold standard. So clearly, from this measurement and many more, we know that the frequency domain system is giving us accurate measurements of hemoglobin oxygenation. They did the measurement on a human as well, had the human go from breathing room air to breathing 10% oxygen, and this is the absorption spectrum that's measured. You can see that the absorption at the lower wavelengths increases, right? If we look at the spectrum, remember the lower wavelengths are more strongly attenuated by deoxyhemoglobin, and because deoxyhemoglobin is going up, we get a higher absorption coefficient. Then, using the spectroscopy, you can convert that into hemoglobin concentrations and see how that changes with this physiological manipulation. Now, looking at the mu-A signal, it was interesting. They saw mu-A was fluctuating in time, right? Their measurement was fast enough that they could see the cardiac cycle, actually. So this is actually mu-A being absolute mu-A pulsating because of the heartbeat, right? So pulse oximetry is looking at the pulsatility of the intensity of light, and then does some algebra and figures out the oxygenation. So now with this multi-distance frequency domain system, they could do something much more fancy. They could actually quantify the absolute absorption coefficient, look at the time series of that, do the Fourier transform. You can see the peak at the cardiac frequency. You can then see how that amplitude changes with inspired oxygen, right? And so if you look at the amplitude versus wavelength, you get a very different spectrum now, right? So remember, let me just jump back. There's this spectrum when we just looked at the total signal, and this looked more like that. It's got that strong deoxyhemoglobin component here, and then flattens out. If we look at the cardiac pulsatility, the absorption at the shorter wavelengths is not as strong. That's because we're measuring the arterial compartment now. The pulsing of blood volume from the heartbeat is only in the arterial compartment. The arterial compartment is almost entirely oxygenated hemoglobin, and yeah, when you do the pulse ox analysis, you see basically from NIRS we're measuring 98% compared to the pulse oximetry of 97%. And when you drop the inspired oxygen, arterial saturation actually only drops a little bit, not much. Okay, let's just skip that. So also, there are fluctuations happening on shorter time scales as well. So this is looking at oxyhemoglobin over 10 seconds, and you can see the pulsatility at the cardiac frequency, but then you also see a slower fluctuation, right? You can see the envelope of oxyhemoglobin, a slower fluctuation. In deoxyhemoglobin, you actually see very little cardiac pulsatility because there's very little deoxyhemoglobin in the arteries. But we do see much more of this slower oscillation. And this was a measurement on the piglet, which had that strain gauge that was measuring respiration. So basically what you see is with inhalation and exhalation, correspondingly we have this modulation of oxy and deoxyhemoglobin. What's happening as you inhale and exhale, it's actually modulating the return of venous blood to the heart. Right? So actually you get venous pooling of blood. So this is a modulation of venous blood volume. Right? So it's like pulse oximetry, but instead of on the arterioles, it's a pulsatility on the veins. And it's happening at the respiratory frequency, not the cardiac frequency. So we can actually distinguish that. Right? So if you look,
is that peculiar to the brain, or a little because of the fixed volume that you have, or what is the rationale for that?
Right. No, that's great question. That measurement was actually on the leg, and it would be fantastic if we could make that measurement on the brain. All right, because physiologically, it's good that I get to delve into this. Physiologically, what we care about is how much oxygen is delivered to the tissue, right? And these NIRS measurements is telling us how much oxygen is in the blood. It's not telling us how much is being delivered to the tissue, right? To figure out how much oxygen is being delivered to the tissue, you have to measure arterial oxygen content and venous oxygen content, and the difference is what was delivered to the tissue. Actually, difference times blood flow. Yeah.
So, what was the noise of that? What was the standard deviation of these? I mean, if you keep on repeating, does the signal fluctuate up and down, or is it like rock steady and in those times?
Okay, wait. I'll come back to that question. I'm still finishing this one. Right. So we need that difference. So if we had a measurement of venous oxygenation, it would solve all our problems. But it turns out, I told you physiologically, as you inhale, exhale, it modulates the venous blood volume. It does another thing. It modulates your heart rate. And as you modulate your heart rate, it modulates your arterial blood pressure, and it's called sinus arrhythmia. And as your blood pressure modulates, it modulates arterial blood volume. So we also get respiratory modulation in the arterial compartment. Right? So this measurement works very well if you're not near any large arteries. Right? And so on the head, we have large arteries everywhere, so it hasn't really worked well on the head. But in the leg, you can find, in muscles, you can find places where it works reasonably quite well.
The modulation of heartbeat is on this 3-second time scale. So
no. So respiration modulates heart rate. Yeah. The envelope of the heart rate is modulating blood pressure, right? So at that slower respiratory frequency, right? And that's what
that's sort of tweaking.
Right. And so then
the stability of
the stability.
of the trace.
I mean, there is physiological drift over longer time scales, but at least for this measurement, we're focusing in on, these lower frequencies. So there is certainly drift, physiological drift, at lower frequencies, longer time scales. But that doesn't impact this sort of measurement. Then there's noise at higher frequencies, but this time course gives you an idea of what the noise looks like. And in my experience, this sort of SNR is very typical. So there's very little high frequency noise there. This also, illustrates the point I was making before about shot noise limited detectors. My noise is really limited by these sorts of fluctuations, which are not actually experimental noise. It's something in the tissue. I don't really know what it is.
You talked about the oxygen delivered to the tissue, where you of course need the oxygenation in the veins and the arteries, but also the blood pressure. So can you tell anything about the blood pressure?
I think some people do, but not with NIRS types of approaches, but there might be some photo-based methods of blood pressure, but I don't really know what they are.
So the question was, could you tell anything about the blood pressure?
Yeah.
Okay. Actually, if you could go back one slide, I have a question. So you say here, I don't know what we're talking about. Are we talking about humans or?
This is human. This is human.
I mean, I've done this experiment where I've fed humans with 11% oxygen. They wouldn't let me go below that.
Yeah.
And they get down to 70%.
On a pulse oximeter?
Yeah.
Really? 70% on a pulse oximeter? It's a good thing they don't let you go below 11%.
Yes.
Interesting.
It takes some time.
Yeah.
And I was bolted to the anesthesiology department in the Radiative hospital at the time.
70%. Wow. I wouldn't want to do that. But I guess it's okay. So you're kind of simulating a little bit the high altitude. If you're high altitude, that's what will happen. If you're there chronically, your SAO2 does drop that low. Yeah.
So I wasn't changing the pressure or anything. I was just changing the.
I know. Okay, but the pressure that will just change what's actually dissolved in the plasma, not what's bound to the hemoglobin, right? So that 70% is what is bound to the hemoglobin.
Yeah.
Which is the majority of the oxygen, anyways. Marie Andrews did these experiments, so why didn't they go that low? Maybe it's because it's only a few minutes, the measurement. Maybe it takes a long time to get that low.
You don't want to bring somebody down to 70% quickly, I can tell you. Okay.
Yeah, okay. Let's keep going. Okay. And the reason that's scary, right? So, normally, we've got 98% oxygenation of our arterial blood because our lungs are so good at oxygenating the blood. The venous side is always like 65%. So your tissue is extracting 35% of the oxygen from the blood. If your arterial is now dropped down to 70%, right, then basically those capillaries, instead of being at around 70%, themselves are now down around 40%, and the oxygen gradient from the capillaries to the cells is less steep, and so the rate at which oxygen can be delivered to the cells now is halved, right? And so you can't support as much metabolic activity, right? So that's why it's, and as a result, cells could start to die, right, if they can't get enough oxygen.
Didn't die, but the subject didn't die.
Well, there were medical students, and they're still alive.
You become more susceptible to ischemic conditions, right?
But I did have one person pass out.
Yeah, I believe it. Okay, so you all right. Back to this time course. So you can take a Fourier transform of this. You see the cardiac pulsatility, and you can see a deoxyhemoglobin, very small amplitude at the cardiac frequency. But if you go to the lower frequencies, this is the respiration rate, and there's the second harmonic, And you can do the same sort of pulse oximetry there and get the venous oxygenation at the respiratory frequency. So they did do this. This was piglet. They also did this on a human, on a leg, and they're exercising. And during the exercise, the muscle has a much higher metabolic rate of oxygen. It's consuming oxygen. And as a result, it's going to extract more oxygen from the blood. And so immediately after exercising, you see the venous oxygenation has dropped, down to 50%. And the muscle takes time to restore its energy balance, and so it takes a couple minutes for the metabolic rate of oxygen to go back to normal in the muscle of the leg. So there's other cool ways of making measurements of oxygen consumption with just a simple NIRS system, using a CW NIRS system measuring the changes in hemoglobin concentration. In this example I'm giving you, they actually used the frequency domain system, and so they had absolute hemoglobin concentrations. So if you do an arterial occlusion, and on the arm is the easiest place to do an arterial occlusion, and if you do it fast enough, you stop all blood flow into the arm, right? And so that's what we see. All blood flow is stopped. There's no change in total hemoglobin, but oxygen is still being extracted from the blood. So oxyhemoglobin goes down, deoxyhemoglobin goes up. And because it was a frequency domain measurement, you have absolute deoxyhemoglobin, you measure that slope, it gives you a measurement of absolute metabolic rate of oxygen.
Sorry,
yeah.
Can I ask a question?
Yeah.
When you're talking about the fast and slow curve, so do we have any protocols that describe that? How fast we should do that, or how slow should we do that?
Well, I know it has to be fast, and I know there's a lot of old literature out there.
We know that it should be fast, but no protocol exists.
Okay.
But is it true that if you do it anywhere, in a university or somewhere else, before you do the experiment, you have to write the protocol, and they have to know how you're going to do it in order to use the funds? At least that's the way we do it. Yeah. So the university has its own big brother looking over to make sure that these protocols are followed and in fact are consistent with clinical practice and so on.
You're absolutely correct, but you see, we are physicists. We are not medical people.
Yeah, but I mean, this one is finally...
And we're not able to dictate them, what to do. But the problem is still there.
But I mean, there's lots of papers in the 80s and 90s.
But no protocols. No standards.
So he's saying standards. I mean, there are dozens of protocols.
Okay,
but I don't think anyone stood up and said this is the standard. So, but actually, it's very simple to see in your data whether or not your protocols are.
No, but what I mean is,
so,
if you say fast, I'm doing fast in Finland, you're doing fast in the States, but who knows, who is slower, who is faster.
So fortunately, what he's referring to, and I didn't say to you yet, if you don't do it fast enough, you occlude the veins. The veins are at a lower pressure, so you occlude the veins first, but you don't fully occlude the arteries. So what happens is blood is flowing in, but it's not flowing out, right? And so if you're not fast enough, the data actually tells you, because what you'll see is that this total hemoglobin is going to go up. You'll see that on the next slide, right? So if we weren't fast enough, we start over and we go faster until we see no change in total hemoglobin, right? And so then that allows you to measure the metabolic rate of oxygen in the arm. So we've been working for several decades now on advancing this method so we can measure the cerebral metabolic rate of the brain of oxygen. And so we're really working on that cuff around the neck. And it's getting better and better, and maybe next year we'll Any volunteers? Just
your team.
So, all right, if you aren't fast enough and you just occlude the veins, you get this increase in total hemoglobin concentration. And if you measure the slope of that, that's telling you the rate at which blood is flowing into the arm, if you're occluding the arm. And so if you know the hematocrit, you actually can convert this slope into a total blood flow. Right? So really cool that you can measure all of these parameters with these simple manipulations.
Can I ask something?
Yeah.
When you say you go so slow that you obstruct one of them, the one flowing out, but not the other, how do you, I mean, you measure a slope here, how do you know that you've actually closed it all completely and not just stopped it halfway, for example?
Well, you can measure the blood pressure, right, which won't give you the venous pressure. Well, venous, actually, so the blood pressure is arterial blood pressure, and so if you want to include the arteries, you have to raise that pressure of the cuff much above arterial blood pressure. Venous blood pressure is quite low. So arterial blood pressure is 120 millimeters of mercury, maybe higher if you're hypertensive, but venous blood pressure is 20 millimeters of mercury. Is very low, so it's very easy to collapse the veins. So, and then what's interesting is you notice the slope comes up, but then it plateaus. So what's happening there? Why is that plateaued?
You should close off the other one.
Because of that buildup, and as flow drops, because it's filled up, and the veins can no longer expand, right? The pressure actually is increasing downstream. And so now at that point, blood is flowing out. So the blood is still flowing in, right, but it's also now beginning to flow out. At least I think that's what's happening.
And then, so if the blood is flowing in, so the blood volume is changed in the tissue. And the optical properties of tissues are changed as well, because they more scattering events, whatever. So, and how that can be, influence the results of the measurements.
So, yeah, right. The scattering coefficient is changing, right? So that's why. So this was a frequency domain measurement. So it was disentangling the scattering changes from the absorption changes. So this measurement handled that. So it's a good point. As the concentration of blood changes, it can change the scattering coefficient, right? And if you're just making a CW measurement, it's possible you could be confounded by that. It's possible. It is possible. And that is something we used to deal with a lot over 10 years ago with the brain activation measurements, because we were assuming there was no change in scattering. But then we did a lot of mathematical analysis, and we were basically able to show that the scattering change you'd get from that small change in hemoglobin was insignificant compared to the absorption change for the brain activation measurements. For these types of measurements, you're inducing much larger changes in the hemoglobin concentrations, and so that may no longer be true, and so you may have to worry about the scattering changes. In this specific data, because it was done with a frequency domain system, it already solved the problem because it separated the scattering changes from the absorption changes. And then back to what I just said, I haven't talked about these data in a long time, but actually, so I said I think the blood is leaking out. In fact, we know it's leaking out from these data, right? Because if this saturated here because blood stopped flowing into the arm, then what would happen is oxygen would still be consumed from the blood, and this oxyhemoglobin would still be going up, right? As more oxygen is consumed from the blood, sorry, I said that wrong. The oxyhemoglobin would go down, right? It would go down because it would be consumed from the blood. So the fact that the oxyhemoglobin has also plateaued really tells me that blood flow is kind of restored to the arm, right? And so and physiologically it just makes sense. It's restored because I've built up so much pressure at the occlusion that it's leaking through the occlusion. But that's why we only measured that early slope. So complex answer to a very simple question.
So if what you're interested in is venous oxygenation, and you can make the venous side pulse and not pulse, why not do that? So you can occlude, make the venous side pulse.
This is why I'm working on this.
But you could do it over a short period of time.
You should volunteer.
I volunteer to do it over a short period of time, right? I mean, maybe not for 5 minutes, but for 30, 60 seconds I could do it.
You'd pass out in 20 seconds.
But if you're doing it pulsating with let's say half a second.
Okay, good idea. Let's try it. You're right, and we don't have to occlude the arteries, so fine. I didn't bring my portable NIRS system.
We'll build you one.
All right.
Once you got a laser pointer.
And we got a detector. Okay. I got maybe a couple more slides. Oh no, maybe I have a lot more. So you said 7 or earlier.
I moved it about 15 minutes earlier.
So 6:45.
So we can do that.
All right. I'll be right on time.
Good.
Okay, so then let me just skip these slides. This is time domain. It's not as interesting. This is more interesting. I saw Igor talking about diffuse correlation spectroscopy. So I wanted to elaborate further on that.
We put it together.
Yes. This was data. This was the first diffuse correlation spectroscopy measurement on humans, and it was on his arm. And was this data your arm or my arm? I don't remember. Well, one of us was the subject, and the other one was running the instrument. So this was the 1995. So NIRS, the classic experiment, was doing that cuff occlusion experiment that we just discussed extensively. So in our Geniode's lab, we had the technology to measure a correlation function. So we wanted to do the classic experiment where we would occlude blood flow and we wanted to see blood flow actually go to 0. So what we saw, so I'll come into the details in a minute about diffuse correlation spectroscopy, just give you the data. Is this out of order? Oh, what did I just do? Do I
just go ahead.
Will it keep recording?
I hope so.
I'll just say yes. I'll say yes. Sorry guys, I made a mistake. Let's figure out what's going on. So it just saved it somewhere.
Yeah, maybe.
What's that?
So this little...
it's recording the break now?
So wherever the sound was recorded, you should see
little
thing in the corner.
Oh, you're right. Oh, cool. You're right. I see that. All right, good. I've never done it before. That's kind of cool. Sorry, I'm just trying to remember. Oh, yeah, now I remember. Sorry about that. All right, we can start up again.
Great mouse.
Okay, so I'll explain the details of the measurement in a minute, but when we did the full occlusion, so 230 torr, oh, I must have been the subject, because you were stronger. You could actually really occlude my arm. So that's way above my arterial blood pressure, so it stopped blood flow, and sure enough, we saw that this blood flow index we measured with diffuse correlation spectroscopy went down to 0. And then as we released the pressure on the cuff, you could see that the blood flow would then return. And then you get what's called a hyperemic overshoot. Because the tissue was starved of oxygen for 10 minutes, the blood flow, well, the measurements were really slow at the time. So we only got 4 data points in those 10 minutes. So the blood flow physiologically just increases to deliver more oxygen to the muscles to help them reoxygenate. So the way you make the measurement, diffuse correlation spectroscopy measurement, is really what it's doing is it's measuring the Doppler shifts imposed by the moving red blood cells. Right? So it's just like laser Doppler blood flowmetry. You now need the coherence properties of the detected light, and as a result you need to use lasers with a longer coherence length. Laser diodes, maybe if you're lucky, are 10 centimeters, typically much less, but the path lengths of light through tissue can be much longer than that. So we need to use longer coherence length lasers, And because we want to see the coherent properties, we want to see the intensity fluctuations arising because of those Doppler shifts, we have to detect a single mode of light. So we use a single mode fiber as the detector. With NIRS, we typically use a detector that's 1 millimeter in diameter or larger. Now if we're using a 5 micron single mode fiber, we're getting 40,000 times less light, right? So a lot less photons. So we have to photon count, and then we're in this digital mode, and so instead of measuring actually a power spectrum of the intensity fluctuations, we just measure a correlation function. They're Fourier transform equivalents of each other. Laser Doppler blood flowmetry is a well-established method, uses short separations between the laser and the detector, even uses a pin photodiode as the detector. They just measure the Fourier transform of the power spectrum of the intensity fluctuations, and that width of the power spectrum is proportional to blood flow. But with this, we measure the correlation function instead, the temporal autocorrelation function. We use these photon counting avalanche photodiodes, which is a really good match. So a disadvantage of a photon counting avalanche photodiode is it has a very small active area, but our detector is a single mode fiber. So, that works really well for us.
So can you just make one comment for the students? Yeah. So when we do the measurements of Doppler, so we do the measurements not in a single mode, single photon, mode, and thus it's like analogous signal, means we have the signal that already averaged. When we use the diffusion correlation spectroscopy, we use like in a single photon fluctuations, in terms of intensity, and this technique is more sensitive for blood flow measurements rather than standard Doppler flow velocimetry. That's just simple technical issues, but they're important for the sensitivity.
So we use now more laser power because we have that single mode detector. So we really push the limits of what we're allowed to do from the laser safety perspective. So it's 60 milliwatts we like to use for this measurement. So let me just skip some of this. This shows it nicely. This is a nice experiment from Turgut Durduran and Arjun Yode on stroke patients. And so this is what a correlation function will look like, right? So basically the signal decorrelates on a time scale of 10 to 100 microseconds. So what that is indicating to us is that essentially the intensity of light that we're measuring with that single mode fiber is fluctuating on this time scale of 10 to 100 microseconds, and becomes completely decorrelated by after a millisecond, right? So those fluctuations are very fast, and those fluctuations are happening because of the Doppler shifts resulting from the scattering from moving red blood cells. So if blood flow is reduced, the rate of fluctuations is going to decrease, and the correlation time is going to get longer, right? So this blue is a longer decorrelation time, indicating a slower blood flow. So in stroke patients, there's this clinical question. Should I have the patient laying down, or should the patient be sitting up? So a healthy brain auto-regulates. So, as blood pressure varies a little bit, the brain can regulate and maintain constant blood flow. But if you're now stroked, maybe the auto-regulation is not working. And if they're sitting up, the pressure's lower and there will be less blood flow, right? Or maybe, if they're laying down, the pressure will be higher and you'll get too much blood flow. So they don't know, should the subject be sitting up or sitting down. There's a very simple measurement. You can now use this DCS system and see what happens. So, in a healthy subject, sitting up and lying down, there'd be no difference, right? So in stroke patients, they basically found in most subjects, let's see, so that's sitting up. So as they went lying down, most subjects, the blood flow actually went up. Right? So that would be what you'd want that patient to have, is a higher blood flow, presumably. But a couple of patients, actually the blood flow went down. and so they'd be better served actually sitting up. Right? So I thought that was really compelling because it's a relatively simple instrument that could just tell the nurse should the patient be sitting up or sitting down to help them get less effects from their stroke.
And when is better to do this cuff?
Okay. Okay, so in the final 6 minutes, I want to tell you about something new, and then we'll get dinner. In our very first measurements, it was interesting. So here's the theory, right? So if you have single scattering, you expect the correlation function to decay as a function of the mean square displacement of the moving scattering particles and the scattering angle. This is Q is the momentum transfer of light, which is just related to the scattering angle. It's a single scattering measurement, so you know what the scattering angle is. Right? In the mean square displacement, you could have particles undergoing Brownian motion, so you have a Brownian diffusion coefficient, or red blood cells are flowing, so you have some speed, some linear displacement of the particles. With multiple scattering, we actually have to consider the number of scattering events, and we have to accumulate all of the scattering angles. And once we do all of that, we can actually solve this and find an analytic form for how the correlation function decays. It still depends on the mean square displacement. So when we made our first measurements on our arms, we expected the mean square displacement to go as red blood cells moving linearly, right? But when we fit the data, we found that actually a better fit to the data was by the Brownian diffusion coefficient. So the red blood cells were somehow undergoing Brownian motion. This made no sense to us. I think we worried about it for a few months. We had no idea what the answer was. And so we said, okay, we're going to just call this Brownian diffusion coefficient a blood flow index. And that was in '95. And still today, we really don't know why the Brownian diffusion coefficient better fits the decay of the autocorrelation function. So
in a way, we consider it's like a translational diffusion.
So
when the flow is good, the red blood cells.
Yeah. So basically, I think so the question is, why is it diffusive motion? And so there was, at least my own thinking, I realized that actually the scattering length in blood is incredibly short. In tissue, it's 100 microns to a millimeter, but in blood, the scattering length is like 12 microns. So you can have multiple scattering events in a blood vessel, right? In a 40 micron diameter blood vessel, you're going to have multiple scattering events. So that made me realize well, actually, maybe what I'm more sensitive to is the relative displacements of the red blood cells, right? Not the absolute displacements of the red blood cells. And so I had this laminar profile, and so there would be relative displacements of the red blood cells, but that displacement would still grow linearly with time, right? It wouldn't grow as the square root of time. Well, then I learned that there's something called shear-induced diffusion. So in blood, this shear induces this, what Igor was saying, this effective Brownian motion of the red blood cells. And where you have higher shear, you have a higher effective diffusion of the red blood cells. So that is a possible explanation, right? So actually the red blood cells are undergoing some shear-induced Brownian motion, right? But which one is it? Is it this linear displacement, or is it the shear-induced diffusion that's dominating the decay of the correlation function? So to answer that question, we did some Monte Carlo simulations for diffuse correlation spectroscopy. They're just like any Monte Carlo simulation that you'd run for any sort of photon migration Monte Carlo simulation, but I need to keep track of the scattering angles, right? And so then I can, if I keep track of the scattering angles and I know the displacement of the scattering particles, I can then calculate what the decay of the autocorrelation function would be. So we created this geometry with translational symmetry, just a uniform array of vessels. We could change the diameter of the vessels, we could change the density of the vessels. For each vessel then, what we had to do was, as a photon was scattering inside the vessel, we had to record many key parameters. We had to record the radial distance of the scattering position, so we could account for the laminar profile of the flow. And we had to keep track of the scattering angle and the scattering angle relative to the velocity vector. Once we kept track of all of that, going back to the original derivation, we could figure out how we could use that information to then calculate what the decay of the temporal autocorrelation function would be. I'll skip that. We had some clever computational tricks for how to do this. So the first question was basically, what is the dependence on scattering? So as we were discussing earlier, this is that similarity relation, where when your photon migration obeys diffusion, you don't actually care about scattering and the scattering coefficient and the scattering anisotropy. You just care about this reduced scattering coefficient, US prime. So I wondered, well, my blood G is 0.98. Does it make a difference if I change G? Because I hate running a Monte Carlo simulation with G of 0.98. It's just computationally expensive. It'd be much better if it was G of 0.9. So we ran it with G of 0, 0.9, and 0.98, or 0.99, with constant μs prime. And when we ran it either with diffusion set to 0 or with speed set to 0, so that we could then compare the decays we got in both cases. So what we see is that the decay due to diffusion is more than an order of magnitude faster than the decay due to the linear movement of the red blood cells. So that shear induced diffusion is really dominating the decay of the autocorrelation function. So that's kind of point number one, right? That this now explains why Igor and I first saw in 1995 that we had to fit the data with Brownian motion rather than with linear motion. Yeah.
No, in your diffusion model, you have a diffusivity in there as well.
Yeah.
And I'm just wondering, you use an essentially one-dimensional kind of approach, but I'm just wondering, what if there is an isotropy? If you introduce an isotropy, then you end up with a tensorial kind of a approach to your diffusion, and that might be a simpler way of explaining this. So
it's true for the Monte Carlo simulation. I had that translational symmetry, which made our computation, just the simulation, much easier. But we placed the source of detector, transverse and parallel to the vessels. The results were nearly identical. There was a difference, right? Because you would expect a difference if you're scattering along the direction of a vessel versus scattering across a vessel. So we did see a small difference, but it doesn't really change the results I'm describing.
No anisotropy. There was no anisotropy, angular anisotropy in different directions.
There was a very small effect, right? But it was insignificant compared to this major effect. Yeah.
I'm not sure if I got this right. If you said that if you do the fitting, you have to appeal to the Brownian motion.
Yeah.
Would that have any impact on the OCT angiography method? So we look at speckle variance and so on. So if you do a model with liquid, static, you have the Brownian motion. Yeah. So you have to do the setting to get rid of this static scattering centers in a different way than if the flow is moving. So that is Brownian motion.
Yeah, now. Great question, and we can discuss that at length over dinner. And I think we need to study that more. But what our simulations have shown, and if Martin will allow me just a few more minutes, or is that okay?
Sure.
All right. What we find is as you go to that single scattering realm, it's the convective motion that dominates the decay, not the diffusive. Right? So that's what it suggests. We need to go measure that, right? But also, I think just intuitively, with OCT, you're going in and out of a vessel. You're really sensitive to that bulk movement, and that what's going to cause a faster decorrelation is it's going to be the linear displacement of the red blood cells, isn't it?
So that's the one which creates that.
For the OCT, yes, but in this realm, because we have more scattering events, it just enhances our sensitivity to the Brownian motion. And one possible explanation, and I want to address this with OCT, and maybe you already have the data, which is the shear is greatest, at the surface of the vessel, where the speed is the least, right? From just a surface area, from a cross-sectional area argument, the vast majority of the red blood cells are near the vessel wall where they're moving slowly but have high diffusion, right? So the prediction would be then with OCT that if you measured a laminar profile and made a measurement such that you could distinguish diffusion from linear motion, we would actually characterize greater diffusion near the vessel walls and less, where we've already measured less speed, right? So that's a prediction that I think OCT could confirm. Right, so let's discuss that more at dinner, and I'll finish 3 slides so we can have dinner. Okay, so diffusion motion dominates, so then we just went further. We said, well, how does it depend on the speed? And sure enough, as you increase speed, the decay rate increases, both for the pure convective case or the total case, which is fit very well with diffusion. So I plot this blood flow index, which is just the decay rate, versus speed. We get linear increases. So that's showing just the expectation that the blood flow index is proportional to flow, right? We want to figure out what that proportionality constant is. So I look at other parameters. So I keep a constant speed, but now I'm going to change flow by changing the cross-sectional area of the vessels. Now what I see, there's a nonlinearity, right? So I'm plotting the blood flow index I get from the correlation function versus what the true blood flow is. And I see that as the true blood flow increases, I'm effectively underestimating it with DCS. So why is that, right? So going back to the theory, the decay rate is proportional to the mean square displacement of scattering particles times the probability of scattering from a red blood cell, right?
What is decay rate?
Decay. The decay rate of the correlation function is proportional to the mean square displacement, but also the probability of scattering from a red blood cell. So it's that probability of scattering from a red blood cell that is causing this observed nonlinearity. Because that probability of scattering from a red blood cell is given by the volume fraction of blood, which is just the cross-sectional area of the vessels divided by the area of the tissue. And it's also given by the ratio of the scattering coefficient of the blood to the scattering coefficient of the tissue. Right? So basically, this is kind of predicting that that proportionality is dependent on the volume fraction of blood. And in this experiment, I'm changing not just the blood flow, but also the volume fraction of blood. Right? So that's where that nonlinearity is coming from. So I could write down, right, so that's where we get here. So when I write down the theory for the blood flow index, it's related to true blood flow, but it's modulated by the diameter of the vessel, as well as the hematocrit of the blood. Right? And so taking into account this factor, I'm actually able to fit the predicted, I'm able to predict the blood flow index quite accurately. So it also predicted if I change the hematocrit, it would change the blood flow index. I'm keeping blood flow actually constant. I'm just changing hematocrit. And I'm able to predict the change accurately. Okay.
So when you change hematocrit, you reduce velocity?
No. We changed hematocrit, we kept velocity the same. Okay. So it's kept the same, but the decay rate decreased. And the decay rate decreased because the probability of scattering from a red blood cell actually decreased.
But if you raise hematocrit and velocity is the same, then blood flow is increasing, isn't it?
Well, how do you define blood flow? Most people define blood flow not as the particulate flow, but as the volume.
The number concentration times velocity.
Well, if you define it that way, then your flow is changed. That's not the way I define flow. So maybe if we define it that way, yeah. So actually, that's a whole another can of worms, right? Because flow, how you define flow is not standardized. people think it is, but it's not. So we can have dinner now. Great, thank you so much for
We'll say while Dave is doing fantastic things in here, my ever-inventive students have found a way to bring the beer to the mountain, as you requested. I didn't tell them maybe to keep the wine back up in the restaurant because I can feel a storm coming in my bones, and so while you could grab a beer and maybe bring it with you, you might not want to hang around too long because it might rain very soon. So dinner back up there, courtesy of Igor and his expert. And thanks to Igor for his patience.
Not for me, so first of all, thanks for Sean who bring the very nice meat, lamb, and thanks for the girl from Moscow who helped to cut the carrots. And you know, without them, without this support, and also to him, who demonstrated the fantastic experience in the cut the tissues. You know. Okay, that's it.
The reason I'm rushing is I think it could be in about 5 minutes.
We did any right and a high standard. And these results are still
we never published that.
We did in Russian.
Oh, that's right. Yeah, that's right.
That's a nice picture of you in a tie. I mean,
and I have those papers.
Yeah, that's true. I remember.
Yeah. But yes, but we possibly should. We possibly should.
Does it right? It's right acoustic.
But I try the one.
There's a second memory stick. Try this one. Because we realized already that there are some problems with format. And I formatted them with two different file systems. The one is NTFS and the other is FAT32. And I hope that one of them is working.
That works.
Great. At least we have one.
I discussed tonight the experiment you asked for.
Yeah.
But they saying like the guys whom I'm working with, they build biologists.
Yeah.
They're saying that that if you're talking about the flow